Hey, how's it going everyone? This is FixerMed. Welcome back to my high yield genetics review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 5 of my multi-part video series covering the entire discipline of genetics in a comprehensive but high yield manner. So, without further ado, let's go ahead and get started on today's content. I'd like to open up by talking about polymerase chain reactions, also known as PCR. PCR is a revolutionary lab technique used to amplify specific DNA sequences in vitro. Denaturation is when DNA strands are heated to separate into single strands. Annealing is when primers bind to complementary sequences on the target DNA. Extension is when DNA polymerase synthesizes new DNA strands using nucleotides and primers. The primary applications of PCR include DNA amplifications for research, diagnostics, forensics, and gene cloning. The primary benefits of PCR are that it's rapid, sensitive, and highly specific to DNA amplification in a controlled laboratory setting. Moving on. Next, I'd like to talk about DNA sequencing. DNA sequencing determines the precise order of nucleotides in the DNA molecule. Sanger sequencing is a traditional method involving chain termination using di-deoxynucleotides, also known as ddNTPs. These produce a series of DNA fragments of varying lengths separated by gel electrophoresis. Next, we have next generation sequencing, also known as NGS. NGS has high throughput sequencing methods which enable simultaneous sequencing of millions of DNA fragments. Main applications of NGS include whole genome sequencing, targeted sequencing, and RNA sequencing. The significance of DNA sequencing includes the fact that it facilitates understanding of genetic variations, disease mechanisms, and personalized medicine. Moving on. Next, we have genetic engineering. Genetic engineering involves the manipulation of an organism's genome to achieve desired traits. Some tools and techniques of genetic engineering include restriction enzymes, which cut DNA at specific recognition sites, enabling DNA manipulation. Then we have DNA ligase, which joins DNA fragments with complementary ends. We have plasmid vectors, where we have circular DNA molecules used to transfer foreign DNA into host cells. The main applications of genetic engineering include production of recombinant proteins, gene therapy, transgenic organisms, and agriculture, aka genetically modified crops, etc. Moving on. Next, we have CRISPR-Cas9. CRISPR-Cas9 is a revolutionary genome editing tool derived from the bacterial immune system. CRISPR stands for clustered, regularly interspaced, short palindromic repeats, or DNA sequences containing repetitive palindromic sequences. Cas9, CRISPR-associated protein 9, is an endonuclease that cleaves DNA guided by CRISPR RNA and transactivating cRNA, also known as tracker RNA. Main applications of CRISPR-Cas9 include precise editing of DNA sequencing, including gene knockout, knock-in, and base editing. Advantages of CRISPR-Cas9 include high efficiency, specificity, and versatility in genome editing applications. Moving on. Next, I'd like to talk about some key clinical considerations. PCR enables rapid amplification of DNA sequences. DNA sequencing reveals the precise order of nucleotides in DNA molecules. Genetic engineering allows manipulation of DNA for various applications. CRISPR-Cas9 provides precise and efficient genome editing capabilities. Some future directions for DNA technology include continued advancements in genetic technologies, which hold promise for applications in medicine, agriculture, and biotechnology. Primary conclusion you should draw here is that genetic technologies have revolutionized biological research and hold immense potential for addressing global challenges and improving human health. Moving on. Next, I'd like to do some questions. Which of the following genetic technologies is primarily used for amplifying specific DNA sequences in vitro? All right, think I gave you guys enough time here. Let's go ahead and check what the answer is. PCR is a widely used technique for amplifying specific DNA sequences in vitro. It allows for the rapid and selective amplification of DNA regions of interest by utilizing DNA polymerase and specific primers that flank the target sequence. PCR has numerous applications in research, diagnostics, forensic analysis, and molecular biology. Therefore, the correct answer is E, polymerase chain reaction, PCR. Let's see why the other answer choices are incorrect. DNA sequencing involves determining the precise order of nucleotides in DNA molecule and does not involve amplification. Genetic engineering refers to the manipulation of an organism's genome, such as introducing foreign DNA into host cells or altering existing DNA sequences. While PCR may be used as a tool in genetic engineering, it is not primarily used for this purpose. CRISPR-Cas9 is a genome editing technology used to precisely modify DNA sequences within living organisms. It does not involve the amplification of DNA sequences in vitro like PCR. Southern blotting is a technique used to detect specific DNA sequences in a DNA sample. It involves transferring DNA fragments from a gel to a membrane and hybridizing them with labeled probes. While it is used for DNA detection, it does not involve amplification like PCR. Remember southern blotting, northern blotting, western blotting, snowdrop. That's a good way to remember those, if you guys remember that from the earlier videos. Moving on. Which of the following genetic technologies is primarily used for precise and efficient genome editing in living organisms? All right, let's go ahead and move on. If you need more time, please feel free to pause the video. CRISPR-Cas9 is a revolutionary genome editing tool derived from the bacterial immune system. It allows for precise and efficient editing of DNA sequences within living organisms by using guide RNA molecules to target specific genomic loci for cleavage by the Cas9 endonuclease. CRISPR-Cas9 has revolutionized genetic research and holds great potential for applications in medicine, agriculture, and biotechnology. Therefore, the correct answer is E, CRISPR-Cas9. Let's go ahead and see why the other answer choices are incorrect. DNA sequencing. DNA sequencing involves determining the precise order of nucleotides in the DNA molecule and does not involve genome editing. Genetic engineering refers to the manipulation of an organism's genome, such as introducing foreign DNA into host cells or altering existing DNA sequences. While genetic engineering may encompass various techniques, CRISPR-Cas9 specifically targets precise genome editing. PCR. PCR is a technique used for amplifying specific DNA sequences in vitro and does not involve genome editing. Southern blotting. Southern blotting is a technique used to detect specific DNA sequences in a DNA sample but does not involve genome editing. That will do it all for this video, guys. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. If not, also would appreciate it if you guys could leave a like and potentially subscribe to the channel for more videos like this in the future. And that will do it for this video. So, FixerMed signing off. Goodbye and good luck studying, everyone.